Okay, so transcript. Okay, now it's I think it's okay. Recording transcript. All right. So this session and along with the next two, I mean, like 2 more days, which include like 6 sessions altogether. I think 6 sessions altogether. Will cover basically what we are trying to do is like preparedness for Med 3. Okay, so as you probably already know, the Med 3 is not really kind of it's not a joke. Actually, Med 3 is it's one of the like the probably most difficult the part of the your curriculum. And so I want you to be prepared before we start the Med 3. So hopefully, so for this course, so I mean for these few sessions, that we will just go over some overview of, say, like a preview of what we are going to learn in the Med 3. Okay, so it's basically we are not teaching you everything about Med 3, what you need to know in the Med 3. So that's not possible for 2 hours. Okay, so what I'm going to go through is an immuno and then a micro part. But before we start, I will tell you what you need to know. I mean, what you need to focus on, and for the for micro part. Okay, so immuno is a big basically everyone has to have some immuno. It's kind of brand new for most of you. Immuno is like brand new, but let me see what happened with my pen. Immuno might be brand new, but I don't know why my pen is not working. Let me just get my pen here. Okay, so I don't know what happened. Is I can only write in my in my finger, which is not good. I don't know what happened with the. So I'll just write. I probably have to write in on my with my finger. So let me tell you what you need to know. I mean, what you need to have. Like you have about a month left, right? So I ask you to do these things before the medley start. Sketchy micro, sketchy micro. Does anyone have problem with the getting the sketchy videos? Sketch micro, you need to do at least 2 times. Okay, you need to watch the 2 times. You don't need to like write down everything. Try to memorize everything in there, but you need to watch this at least twice before the medley start. Okay, micro, but sketchy. Originally it was like the micro, but now they became kind of big, so they added the farm, and they made it. It is in the mega file. Yes, good. So they made it in the. They made they added the like farm. They made peso. They made IM and that kind of stuff. But I heard that the other parts are not very useful, but the micro is really important, very intuitive, and especially people who have a hard time in memorizing. I have I'm the worst memorizer in the world. I cannot memorize anything. Really, but this thing just really kind of helps you to the recall things. Okay, so sketch micro along with sketchy among the sketchy farm, only antibiotic path, sketchy antibiotics. Okay, so antibiotic part is not that big, but it's a very, very useful. Okay, so sketchy 2 times. You need to watch all of this. Not it doesn't take very, very long. Okay. Also, another thing I highly recommend is Pesoma. Pesoma, the chapter 1 through I think it's chapter 1 through 5 is basically covering all the fundamentals of the pathology, as well as the like the red blood cell and white blood cell stuff. So this chapter only you need to like watch 2 times. Okay, and the as you going along with the semester, you just watch that particular section. For instance, if you you will do the cardio, but you will do the muscular skeleton. Watch that the muscular skeleton part in the sketch. I mean, the Pesoma. Okay, so Pesoma is a big thing. Okay, you need to know. You need to have at least these two. At least these two. You need to have before during the break, basically. Okay, before the semester start, you need to have this. You don't have to write every details. Don't worry about every details. But by having this sketchy, micro, and antibiotics, Pesoma 2 times, chapter 1 through 5, that will just get you ready for the semester. Big game. Okay, so this is a very, very important. And then if once you get ready for this the sketchy and then the Pesoma, you are good to go. All right. So I'll just erase this thing, and my the for some reason my pencil is not still working. I don't know what happened. So I'll probably have to the write on with my fingers. What is it here? Oh yeah, now it's working. Okay. Right. All right. So there are immunology is probably I mean, at least for me, it was new to me. Okay, I didn't have any immunology before. I mean, because of my background is not the bioscience, but do you learn immunology when you're in like in the college, in the bachelor's, or in the high school? Do you learn immunology? Anybody learn immunology before this course? Yes. Oh, when do you learn? Undergraduate. Oh, I see. I wasn't on the bioscience major, so I had no idea. Okay, so if you had the immunology background, I mean, if you learned this like immunology before, that'll be very, very helpful. But for me, everything was like brand new. So, it's a kind of, immunology was like very, very like different language for me. But anyway, so basically there are 2 types of immunologies, immunities, innate and then adaptive. Okay, innate is basically think about innate as a blood. What I mean is that the blood vessels are everywhere, right? Blood vessels are in your like outside world, and then this is inside your body. Then blood vessels are everywhere. So blood vessels, including, I mean, there are some kind of the physical immunities like skins and all those stuff. Those are not really a big thing. Okay, so that's very, very the intuitive ones. People they don't test you that kind of stuff. That's too easy for you. Okay, so what they are asking you, it will be in the what will be in the exam will be something to do with blood. What I mean is this blood in terms of immunity, the innate immunity is blood has a many blood because the blood is like continuously circulating, right? So what's in the blood is the with the non-specific immunity. what this means is that these guys, the innate immunity, the immune cells will just kill everyone, which is not yourself. So, for instance, example of this is like neutrophils, eosinophils, basophils, all basically this is a granulocyte. The reason that is called granulocyte, if you look at this, all these things, do you see this like if I blow up, how can I blow up? If I blow up here, do you see this like the this where is it? Yeah, you see this granules here, granules here. There are many granules inside of this thing. That's why it's called granulocyte. Okay, so again, the digesting the whenever I see new terms, I always try to digest the terms. So granulocyte, cell, granule. So granule. So basically fine particles, small particles. Okay. So whenever you see the terminologies, new terminologies, try to digest that, the break down that word. Okay. So granulocyte, all those are granulocyte, and then monocyte, and they have like different names. Okay, monocyte. Okay, let me because this is important. I'll just write down here. Monocyte is same as macrophagy, same as Langerhans cell, Langerhans, and then Cooper cell. You already know what Cooper cell is, right? Cooper, I think. Cooper cell, and what else? The eosinophil. I mean, no, the histiocyte. Sorry, histiocyte. All those are same thing, basically same thing. Okay, so only difference is that when it is in the blood vessels, it's a monocyte, and then when it comes to the tissue, it becomes the macrophagy, and then activated, and then when it becomes in the skin, the Langerhans cells. But don't worry, and then also alveolar macrophages. Don't worry this thing. They are all basically same thing. It's the same cell, just a name changes. Okay. So all those just know that the all those are referring to the same thing. All right. So monocyte, so dendritic cells, all those are basically the innate immunity, innate immune cells. Okay. So now one of the each of these cells have some kind of uh, cluster of the differentiation, something like CD markers. Okay, so CD marker is the one that every the tissues or every cells in our body has a some kind of receptors in our body, right? In our cells, right? So these cells, some they have a like unique kind of name tag. Okay, so one of the name tags. So bone marrow has their own the name tag. Everything has a name tag. Okay. Uh, One particularly important is a natural killer cell. So CD16 and then CD56. That's a very unique in the natural killer cell. When you see the CD marker, 16 is high or 56 is high, then you will see, they are the high in natural killer cell. One of the easy way to remember CD16 and then 56 is think about the age 16. The killer girl, right? Fine, really sexy, the fine girl, right? That's the wow, she's killing me. She's so hot, right? So that's 16 years old. And then when the woman becomes 56, is literally killing, right? She's literally killing you, right? So 16 and 56, okay? So I think Dr. B, who was whatever her name, I forgot the name. She will explain this in detail too, okay? So that's what you need to know, all right? And then adaptive immunity. Adaptive immunity is a little, it's a kind of second line, the defense. So it's second line doesn't mean that it's the like weak immunity. It is actually so more specific, and then the more the stronger. say a relationship usually. Okay, so what specific means that it has a memory, and then whereas the innate immunity, I mean the primary innate immunity, no memories. So basically, innate immunity means like whoever comes in, which is not me, then I will kill him. Okay. Adaptive immunity is very specific to kill those pathogens. Okay. So one of the very important thing is antibodies. Antibody means anti, and then body is a short form for basically here is like pathogen. You can just think of it as an antibody as a pathogen. So what this means is that whenever there's something uh, need to be killed, antibody will just just tag. Antibody is like a tagging this pathogen here. Then antibody tags you, tags the this the pathogen, and then it will send the signal to the like some killer cells. Killer cells, the actors, the like neutrophils or the B cells, T cells, not the sorry, not the neutrophils, B cells or T cells, or those cells will come in and then kill the pathogen. Okay, so antibody is not really directly killing the pathogen because antibody is very tiny the just peptide. So only few proteins in the antibody itself. Okay, but pathogens is huge. Okay, usually. So antibody is just tagging the pathogen, and then B cells or T cells will come in, and then okay, oh this the antibody was the tagging it here. Okay, so I have to kill them. Okay, so that's what that is. Okay, there are like 5 types of the antibodies, like the immunoglobulins, immunoglobulin, right? So this is kind of globular shape, but if you it's a little bit misleading because it's not really globular shape like a globule, like the circle. It's not like a spear shape. It's actually it's kind of this shape. Okay, so but this immunoglobulin is a little bit about the misnomer. Okay, all right. So, and that's what that is. And then if you have enough time, then spend more time in the BDJ recombination. I wouldn't recommend this, spending any time on not even 1 second to do this thing because this is very low yield and then almost non-existence in the step 1 exams. Okay. So just whenever she probably spend a little time in the BDJ recombination process, that process probably you will be better off studying something else other than this topic here. Okay, so but just know that there is the recombination process. What you need to know is though when you make the immunoglobulin IgG, for instance. Okay, IgG. Okay, let me just ask you this question: human and then the bacteria or any pathogen like a virus, which is more advanced, which is more smart. Many people think that human is more smart, right? But actually, that's not true. These guys, the the organisms, very smart. They are super smart people. I mean, super smart things. So, whenever we make some other drugs, or we make our body makes antibodies, then they keep changing. They are keep mutating. So to avoid uh, the our killer the weapons, okay. So what's funny thing is that the reason that the virus or bacteria are smarter than human is not because they are well educated, because they are so dumb. It's a little irony because what happens is they are so dumb, so they make mistakes. So in other words, mistakes means error. So what that means is that when you make the DNA or RNA and those things, they make mistakes. We in the human don't make mistakes. Even if you make mistakes, there's like the mechanism that remove that the erroneously generated DNA, RNA, or nucleotide, right? But so the these viruses and then bacteria don't have that kind of mechanism. So it will just make mistakes. Then that mistakes is the directly reflected to the RNA, DNAs. So their uh, RNA, the like made in say the 10:27 a.m. December 13 is different from RNA that made in 10:30 a.m. So very, very many mistakes, and then the mistakes is directly reflected in the RNA and DNAs. So there, even if we make some like antibodies or some we develop some drugs to kill those bacteria, that bacteria is not the same when you made in a different time. So they have a like very smart way of the overcome the weapons. Okay, and so that's something you need to know because it's all because of the they call it is antigenic variation. Antigenic variation is mainly due to the due to dumbness. Was error due to error. Okay, this random error will make the antigenic variation. Very important concept. Okay, all right. So basically, the adaptive responses are done by B cells or T cells. So when you think about adaptive, B cells and T cells. Okay. So another name for that B cells and T cells, B cells, T cells are lymphocyte. Yeah. Lymphocyte, when you call lymphocyte, it's the B cells and T cells. The I even like send the inquiry to up to date the editorial people that leukocyte, right, about this. Leukocyte actually is kind of misnomer because leukocyte is a white blood cell, right? White blood cell is kind of misnomer because leukocyte contains the neutrophils and all kind of white blood cell which is not red blood cell. So like leukocyte, neutrophils, basophils, and eosinophils, macrophages, and the B cell, and also B cells and T cells. But these are the not the we don't call it the lymphocyte. Lymphocyte is only B cells and T cells, and then sometimes they include the natural killer cell too. So these are the lymphocyte, but these are not the lymphocyte, but they all both are called leukocyte. That's kind of little misnomer. So instead of calling this the leukocyte, we may just call this the lymphocyte, and then this maybe just give something else. But they still call this white blood cell, white blood cell. That's a little bit of a misnomer. So that's a little confusing point. Okay, so just let you know that. And there's a like antigen presentation. So what happens is when the the pathogen, new pathogen comes in, then antibody is basically in the blood, okay? In the blood vessels. In the blood, it will just circulating all the way, always circulating, and then what happens is there's like some antigen presenting cells. Okay, so that includes dendritic cells, macrophages, monocyte, and this. Macrophage and monocyte basically same thing. So don't get confused. Oh, macrophage and monocyte, there are two different cells. No, they are basically same thing. Okay, so cross out. Just there's name changes, but they are basically same thing. So same cells. Okay, and then also some B cells. Those are APCs, antigen presenting cells. So what happens is when the new pathogen comes in, right? Then these cells will like see these cells. this the new pathogen, right? So what they do is like they will send the signal to some some headquarter, right? Their headquarter. Now then problem is this cell, how can they present the this the there are some new pathogens coming out. So the way they do is they don't they cannot, if sometimes this is like too big or something like that, right? So what they do is they just pick only part of this the pathogen, which is called epitope. 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 Okay. Epitope is basically part of the peptide. So it's a small part of this pathogen peptide. Okay, so small the like peptide. Okay, so then you take that and then present to the some the lymphocyte headquarters. Headquarter in this case means like it present to the B cells. The B cells can you know generate it, differentiate to the plasma cells. The plasma cells makes the antibodies, which is IgG, IgM, and that kind of stuff, right? The antibodies. Okay, or it can send the signal to the like present it to the T cells. So T cell can do other type of the immune responses. Okay. So it's like when you see something, some like the gangsters comes in the town, then you don't like bring that gangster to the police station. You just oh take pictures of the gangsters. Okay, the gangsters are coming in to the police station. Okay, so that's like the same analogy. Okay, all right. Then okay, so there's like the inflammation. Okay, inflammation has like the inflammatory response is you already know this, right? When you have inflammation, so you have like the ruber is red, and then this is a dola. Dola is like the in Spanish is the pain. Tumor or is the kind of the I think it's the tumor is kind of the edema, that kind of stuff. Okay, so those are like the acute inflammatory signs. So when the new pathogen comes in, right? So pathogen comes in, it will be detected by this thing, what is it? Toll-like receptors or something like the PAMP, PLR, yeah, PLR. That's the word that I was looking for. PLR is the PLR means like pathogen pathogen something respond receptor or something like that. So this is a kind of antenna. Okay, so this is a PLR, which include the which PLR is antenna that like recognizes the like PAMP and DAMP, which is like a pathogen associated the mucoid, something like that. But so that will be detected. Antenna will detect those things, the this PAMP or DAMP, and then that will send the signal. So when the receptor comes in, this is like the antenna. Okay, that's PLR. PLR is antenna. Okay, and then detect these things, and then send the signal to the body, and then that signal will the initiate many the responses. Okay, so that will be called immune response. Okay, and then there are many ways to protect our body from that signal. So we'll make the the inflammatory cytokines, IL-1, IL-2, and that kind of stuff, or some bradykinin and all those things, complement, and all those things will be the immune like orchestrated. Okay, so we'll go through this. Basically we will learn how this will work. That's the basically whole everything about the immunology. Okay. So if we know this, the response, then you're done with the immunology. Okay. All right. So the one big thing, very important thing, um, I mean, I understand this is important, but I don't know why they are so important and so popular topic in the exam. Is maybe one of the reasons is like this. That's my understanding. Is that because this process is important. That's one thing. Another thing is that the immunology is very, very complicated. If we want to know everything about the immunology, that's probably not possible in your lifetime. So, but this process is well known and well established truth. Okay, well established fact. So this is because people, already find out this mechanism exactly. So that's why this is probably the one of the popular topic in the exam. Okay. So anyway, so this topic will be on the exam. I can guarantee. Okay, if it doesn't come out in the CAS, it will come out in the step one. And then if it still doesn't come out to step one, maybe you can blame Alex and Sunny. No, just kidding. Okay, all right. All right, but this will come out in the exam for sure. I can guarantee. And then they will probably not only one question, probably few questions, just changing the vignette of the question. Okay, so all you have to know is this mechanism very, very carefully. Okay, so I'll just the explain this to you. Neutrophil always just in the blood vessels, you will always see the neutrophils, always. Okay, so neutrophils always in the blood vessel. Now, when you have like some kind of injury here, okay, or some other pathogens coming in here, bacteria or the viruses, whatever is coming out here, then that the neutrophil has to come out, right? Because of the it will send the signal to the like this will the antenna, the PRR will detect this, so something new gangsters are coming in out into our body. So it will send the signal to the blood. That blood circulating, and then it will be the detected by signal to the neutrophils. So neutrophil has to come out. Okay, neutrophil is going to come out. The way the neutrophil comes out is kind of like think about speed bumps. Okay, so when you go to like highway, no problem. But when you try to the going to your parking garage, right? So you have to slow down first, right? So slow downing is the is called rolling. So margination simply means that it will first blood vessel here, it will first go to the side, kind of into the side of the kind of sideway of the blood vessel. It it was in here. This is blood vessel. Okay, so it was kind of going to the near the wall. Okay, that's the margination. Margination, and then all this is all caused by PRR. PRR antenna will send the signal that will the cascade all these kind of things. Okay, so margination first, then after that there is like a little speed bump. This is a speed bump. Speed bump is done by the E-selectin and then the also ICAM. Okay, so so you need to know this thing, these names here. Cyanotic Lewis, that's a kind of receptor in the neutrophil. And then also the E-selectin is in the endothelial cells. So that's something, which one has which one? That's important. Okay, E-selectin, endothelial cell. The acid, which is in the neutrophil. Okay, and then the ICAM is also in the endothelial cells. Those two are in the endothelial cell. Let me just write in here. This one, endothelial cells. Endothelial cells. I think I always explain this thing. Endothelial means that endo means blood. Okay, so blood wall has this and this cells is a blood vessel. That's the endothelial cells. This is the endothelial cell. Okay, which is different from epithelial cells. Epithelial cells means not blood. which is in the tube, like the proximal convoluted tube or whatever the tube or the bile duct, that tube has a wall too, right? In that wall is covered by epithelial cells. But when it's the comes to the blood, it call it endothelial cells. Okay, so endothelial cells, and then this is the blood basically. Okay, and this is blood vessels. Endothelial cells has this the receptors, which is E-selectin and ICAM. Okay, then uh, the like this these chemokines from the blood by the antenna PRR will the do some like this kind of actions, and then they will send out the signals like this the chemokines. Okay, so chemokines means like chemo means a chemical. Then kines it means some kind of the materials that carries the chemical materials. Okay, so this where is it? Okay, so that will bind to this the like neutrophil bind to this the E-selectin, and then it kind of slows down. So slows down, but doesn't stop. Okay, but and then the when see this thing not attached yet, right? When it slows down, and then as soon as it slows down in few maybe centimeters or something, that after that it slows down, then only after that there is a like a ICAM, which is ICAM. I means integrin. Okay, integrin will bind to this the what is the PE-chem. Okay, integrin ICAM bind to the PE-chem. Okay, and then let me see PE-chem here. So let me see. Oh, sorry. It's not PCAM. It's bind to it's bind to the leukocyte. LFA is like a kind of receptor. Some I forgot the name of it. It says a leukocyte. The something. I'm not sure. So LFA receptors, and then that bind to the ICAM, and then only after that it will try to it stops. Like now it's stopped. Okay, flow down, and then margination is going to the side of the road, and then it rolls, slows down, and then it binds too tightly, and then now it's stopped in here, and then now it's ready to roll, and then it goes through this the between the endothelial. It's kind of it is a most of the time this the endothelial cells are tightly the connected with the tight junction, so the blood don't leak. But there's always like a channels, right? This thing, channels or receptors, that kind of things, right? But in this case, as you look at see here, it's not going through the channel or the receptors. It will go through the between the endothelial cells. Okay, it will become fenestrated, meaning that these two endothelial cells will become fenestrated. It will just open the doors, open the like they are tied together, and then now they just okay, the moves to the side, and then move to the side, so that the neutrophil can come out, because the neutrophil is a big, huge. So this is a cell, this is a cell, right? Even bigger than endothelial cells. So it cannot go through the channels. So it has to be the endothelial cell has to be spread apart so that the neutrophil can come out. Okay, so that's called transmigration. Okay, because it like changing the location, the migration, changing the location from blood vessel to the tissues. Okay, so this process is you need to know like everything about this thing. Okay, so this will be very popular topic. Okay, so it's not easy, not hard problem, not hard, but it's very the popular topic, so you need to know. Another thing, very important thing, is that when the when when the neutrophil comes out, what's going to happen is the neutrophil will say pathogen here. Neutrophil is a cell. Pathogen sometimes it's like the small bacteria, or sometimes like some virus or whatever, right? So what happens is that this the neutrophil will try to eat up, and then the eat up by phagocyte, phagosome, phagosome. Okay, and then there is also the death chamber, lysosome. Lysosome is a death chamber. Say like the electric electric cushion, the chair. Okay, so what happens is this thing will phagosome will first eat up this the pathogen inside by endocytosis, right? Eat up, and then there's a like a lysosome inside of this the inside of the neutrophil. There's a lysosome which is a kind of death chamber. Okay, so this has to be uh, together, fused together, so forming phagolysosome. Okay, then in order to the organelles to attach together, combine together, what do you need to do? You need a microtubule polymerization. This is where what you learn in the med one, it will come back. Okay, microtubule tubule polymerization. So you know that the microtubule will form polymerized. It will just polymerize here, and it's kind of the rope. Okay, and then it will just pull each other. Okay, so pulling each other, but that's so then the pathogen, and then goes into the rise they combine together, and then it goes to death chamber. Death chamber has a lot of ROS, mainly ROS. ROS means the reactive oxygen species. So basically, the oxygen is like a kind of toxic thing, right? So it will generate the free radicals, and the H2O2 and OC something minus something like that, all kind of things, right? Those will is all free radicals, and then to kill the pathogen. Okay, so that's how it works. Now then, the question is if there's a problem in this process. That's why this process is important. Okay, one of the problem can be this one, LFA, the deficiency. So, like leukocyte adhesion deficiency. So this one will not the adhere. So this is not there. Then this process, the combining ICAM to the LFA is not, it cannot be done. Okay, if it's not if it's not there, if it cannot be the cannot be attached, so it doesn't stop. What's going to happen? So neutrophil cannot come out of the blood vessel, right? So if you measure the neutrophil level, so do you think neutrophil level will be high or low? Remember, always measuring something in your body always happens inside of the blood vessel. Okay? You cannot measure anything in intracellularly. You cannot measure anything other than blood vessels. Even very hard, you can. There's a way to measure the outside the blood vessel. Some if there's something in there, but that's kind of that requires some special technique. Okay, special equipment. So when you measure something, always blood vessel. Okay, so neutrophil levels in blood, is it going to be increased or decreased? If that the this stopping cannot happen, what do you think? Is it going to be increased or decreased? Neutrophil levels. If he has a LAD, so LAD means that the addition is not there. What do you think? Anybody can answer? Is it going to be neutrophil level will increase or decrease? when there's a the say the pathogen invade, so inflammation. So what do you think that the LAD will be? Okay, can anybody answer that? You can unmute or the answer, or you can just the type in the chat. Yes, yes, it will be increased because the remember the antenna PRR will send out this thing, the cytokines. So okay, yeah, there's an inflammation. There's a new guy coming in, some invaders coming in. So send out the neutrophils, and then neutrophil comes in. So neutrophil will be recruited, or neutrophil will be new neutrophil will be made, and then from the bone marrow, and then it will just come out many neutrophils in here, but cannot come out because it doesn't stop. It just rolls in here, slows down, but doesn't stop. So let me just ask you one more thing. Although this is not very clinically important, but neutrophil speed. What do you think? Neutrophil speed will be high or just normal or the slow down? Neutrophil speed will be slowed down, but cannot come out. Okay, slow down. That's important. Okay, because you don't have any problems in the margination or rolling. No problem. Only problem is you cannot stop. It will just roll, keeps rolling. So the neutrophil will be just maybe many neutrophils will be around here, covering the some blocking the some of the endothelial cells areas. Okay, but these many neutrophils, but few neutrophils in here inside of the blood vessel, like in the center part of, no, not many other in the center part, but in the marginal part, in the like side of the road. Okay, so that's the important thing. Okay, and so what happens is in that case you cannot heal the wound. If there's a wound, if there's a cut, then neutrophil has to come out and the fight with the bacteria and or some the inflammatory materials. But I mean, there's there are some pathogens. But the neutrophil cannot come out of the cell. So a lot of neutrophils inside of the body, I mean inside the blood vessel, and then those neutrophils are very kind of slow down. This part is okay. This part is a problem. Okay, so and then another one very unique thing is that umbilical cord delayed detachment. That's a very important thing. Because I think I read the article for this one because of the reason this is because of umbilical cord when the mommy here, the baby here, and then umbilical cord. This umbilical cord was when you it's not like a the kind of is when no more the blood vessels. I mean, no more the connected to the placenta, and then blood supply is the not there, right? So also kind of ischemic damage, right? Ischemic damage. Then that's supposed to the initiate the inflammatory process because of ischemic damage. So cells are damaged, then inflammatory process supposed to initiate it. Now then the problem is this the neutrophil cannot come out. So neutrophil should come out and then to start the initiate the inflammatory process, but it's not there. So that's why this is a kind of delay. But no matter what the real pathogenesis is, you have to know this is very unique in the LAD. So LAD, think about umbilicus, and then neutrophil high. Okay, two things you need to know. All right. Okay. So another question, which I haven't seen this kind of question, but I explained this to you. So if you this is true, but neutrophil, yes, true, neutrophil level be high. But if there's a like measure that the say this was blood vessel, neutrophil here and then neutrophil here, right? If there's a measure to majority only in the center part of the neutrophil. I mean, in the blood vessel. So through the only the center part of the neutrophil, I mean in the blood vessels, then neutrophil level will be low because all neutrophil will be in the side of the blood vessels. Okay, neutrophil here, neutrophil here, neutrophil here. Okay, all right. So something you need to know. But that's a kind of side because there's no I haven't seen that kind of question, but it is that's the case. Okay. Now another question, another thing is that there's a celiac Higashi syndrome. So this one is a problem is this thing, microtubules supposed to connect this the phagolysosome. If this thing is not there, the I mean the pathogen is coming to here. Pathogen is inside of the neutrophil, but neutrophil cannot kill this guy because it's not connected. So only the neutrophil eat up the bacteria or virus, but it doesn't. This bacteria cannot be killed. It's living inside of the bacteria is inside of neutrophil. So that's what happens in here. So in this case, so this is only one example here, but there are actually other the bacteria or pathogens that disenable, it will inhibit the phagolysosome, the attachment, phagolysosome forming. So, for instance, you will learn this in the microbiology. For instance, the Botadella, the 100-day the coughing, Botadella pertussis. Okay, Botadella pertussis is one of the bacteria that inhibit this forming, phagolysosome forming. Okay, also TB, also histoplasmosis. Those conditions, it will inhibit phagolysosome forming. Okay, so that's something you need to know. In here, the celiac Higashi syndrome is due to the microtubule defect, and then this one is due to the bacteria or due to the other pathogens that will inhibit this process. I don't know exact mechanism how they inhibit it, but they maybe they send some kind of the chemokine or something like that, so they will just inhibit this function. Okay, that's something you need to know. All right. So I mean, another thing is that this is kind of the that's due to the Higashi celiac Higashi due to least gene defect. Okay, something you need to know. All right. Oh, another big important thing. Okay, giant cytoplasmic granules. Because what if you see this in the microscopy, then you will see like granules become large because bacteria is inside. It doesn't kill. It's still inside of the neutrophil. Okay, so that's what happened. All right. Now then the chronic inflammatory process. So I think the Alex will explain in the second session for the pathogenesis. I mean the pathology part. So I will just leave it her to explain this one. Okay. One important thing. Let me see. Yeah, one important thing is in here. Okay. Yeah. In the chronic inflammatory process, one important thing is that when the like some other when normal bacteria comes in, normally macrophagy comes in, macrophagy eats the bacteria and then kills the bacteria, and that's it, right? And then when bacteria kills neutrophil kills the bacteria, what's going to happen is the bacteria will die, right? So then the neutrophil also neutrophil is kind of suicide bomber. Suicide bomber. Yeah. So when neutrophil kills the bacteria, and it will die. Okay. Whereas macrophagy is normally long lasting. So normally just kills the bacteria, but still do their own job. for a long time. So macrophages. So it's big. Macrophages becomes huge. Okay. So neutrophil, whereas neutrophil it's only one of maybe few bacteria, and then it cannot eat anymore, and then it will suicide. Okay, with the bacteria. So when the bacteria suicide, so with bacteria, what is this material? This material you learn pus, right? Pus is this is pus. Basically, definition of pus is this thing. So pus, if you look at the microscope, then pus you will see the dead bacteria, and then dead neutrophil. Okay. So now what happens is so there are some bacteria or some other pathogens that neutrophil or macrophages don't know how to handle. So for instance, the typical one of the good example is a TB. You know TB, right? Tuberculosis. So what happens is a TB bacteria comes in. It's a bacteria, and then comes in, and then macrophages and neutrophil first to try to eat up, but the neutrophil don't know how to handle. So now then in 4 days later, macrophage comes in. Macrophage try to eat up, but this thing is not like it's a very very strange thing. The macrophage cannot eat. So they fight, try to kill this bacteria. But the many times, oftentimes that the bacteria cannot be killed. So some bacteria, macrophages can kill some of them, but not all of them. So there is some bacteria die, and then macrophages, what macrophages will do, and some also the neutrophil already died. So there are some neutrophils, dead neutrophils, and then the dead bacteria. But there are also some other live bacteria which the macrophage cannot kill. So then what macrophages is programmed to do is that, oh, I don't know how to handle this thing. Then bacteria will wall off this these things that inside of this thing is dead neutrophil, and then dead bacteria, and importantly, live bacteria, and some live, I mean the dead macrophages. So the way you the way the macrophage handle is this: macrophage try to eat up macrophages here, and then eat up this bacteria, and then some of them cannot be eaten, or some of them cannot, even if it's eaten, but cannot be killed. So macrophage will just ask his friend, another macrophage, one macrophage, two, and then yeah, let's just work together to kill this guy. So I cannot handle. Okay, so let's take one more guy. So it will be macrophages will be big. Like macrophage has like special functions, so they can combine them together. So macrophages, many macrophages will work together, forms multinucleated giant cell. It's basically macrophage team. Okay, so this is a multinucleated giant cell will formed. That means whenever you see multinucleated giant cell, some new bacteria, new pathogens in the block that macrophage cannot handle. Okay, so macrophages will form a big team. Okay, like a league of teams, and then they will. So you will see inside some dead bacteria as well as the live bacteria. Live bacteria is important. Okay, so they will form multinuclear giant cells, and also they will just wall off. Okay, so what happened here is macrophages here, macrophages wall off this whole area. Okay, this area. So this is inside like I said, very important. There's a live bacteria inside. Okay, so this is called caseating the granuloma. I'll write in here because it's important. Oh, uh, caseating granuloma, TB. Okay, TB, and then some other conditions like histoplasmosis and that kind of thing. Okay, then it will form this the granuloma, which is a caseating, means that there's a live bacteria inside. Okay, so what happened is there is the chemokine called TNF alpha, okay, which is secreted by I think this is the macrophage TH1 cell will secrete the TNF alpha, and then TNF alpha's function is to keep this the granuloma stable. Okay, so make it stable granuloma. Now, what happened if the TNF alpha is not there? What's going to happen? It's not. It doesn't keep this thing stable. So, in other words, this will be broken, right? If it's broken, that's what's going to happen. Live bacteria will come out, right? That will be causing the latent TB. Latent means late TB, delayed TB. Okay, so this thing, why it's not stable? Because of TNF alpha is not functioning properly. Why is it happening? Most of time is due to the drugs, the TNF alpha inhibitors. Why do we take these things? Because of to treat other conditions like rheumatoid arthritis or some other chronic inflammatory SLA or something like that. I mean SLE or rheumatoid arthritis or other conditions that try to kill this, treat this the conditions. We take the TNF alpha inhibitors. On examples, the natalizumab, and some other conditions, some other drugs. Okay, so those kind of monoclonal antibodies and other drugs that inhibit TNF alpha inhibitor, TNF alpha. Okay, then TNF alpha is not functioning, and so it will not be stable anymore, and this will be the granuloma will be broken, and then live bacteria, live TB particularly, come out, and then you will see the latent TB. Okay, so this is very, very, very important thing you need to know. All right. So this will be on the exam for sure. All right. Usually, the question Binya is like this. So Binya is the some like middle-aged woman has a rheumatoid arthritis, and then take some the rheumatoid arthritis medication. They don't mention the name of the drug. Okay, so they take the rheumatoid arthritis medications, and then like few weeks later, he was coughing, and he cachexia, and then the weight loss, and then what's the cause? The cause is a rheumatoid drug, adalimumab, the natalizumab, those things that disable the TNF alpha, and then that inhibit the TNF alpha. That will disable the granuloma stabilization. So the granuloma will be broken down, and then wall is broken down. So the TNF alpha, I mean the live TB will come out, and then that's the problem. Okay, it's a very, very important thing. Whereas non-caseating, that's I just explained the caseating. Although caseating is much, much less than the non-caseating, because other than TB or the histoplasmosis, most of time granuloma is non-caseating, but non-caseating is not is less question in the question than the caseating, because caseating is more problem clinically, and then non-caseating is not really a big problem in terms of the like the side effect of the drugs or that kind of things. Okay, so this is, although this is less common because only in 2 instances mostly, but it's more question in the problem the exams. Okay, the non-K shading is more common, but it's a kind of the non-K shading simply means that no live bacteria inside. Okay, that's non-K shading. K shading, live bacteria inside. Okay, that's what you need to know. All right. Now this thing, the pro-inflammatory cytokines. Cytokines simply means like cytokines, something like a chemically some kind of signal. Okay, signal. Okay, this signal, what you need to know in the is in the first aid mnemonic called the hot T-bone steak. Okay. Okay, so that's something you need to know. Hot T-bone steak. So this simply means that IL 1, 2, 3, 4, 5. Okay, so I think 6 up to 6. Okay, so hot means heat, like fever, right? So IL 1 causing hot fever. T uh T is a little bit of a T is like IL 2. IL 2's function is proliferate. Right. So it will just make more T cells, more T cells, more T cells. Okay, so T, and then IL-3 is I think IL. What happened is I think I had T-bone state. Okay, the they make it. They try to make it the mnemonic for this. I think that's what it is. So, but IL-3 is okay. But you can use the different kind of interpretation. But I think the best way is probably this way: hot, the hot growing T-bone steak. Okay, so add the growing here. Hot growing T-bone steak. Steak. Okay, so hot means IL-1, and the growing, so proliferating, that's IL-2's function. T, so every CD4 cell, all CD4 cell has IL-3. Okay, and then IL-4 is IL-4 is eosinophilic, the eosinophil, the recruiter. Okay, and then IL-5. That's IL-5, and then IL-6 is I think is also causing the fever too. Okay, right. So that's something something it's kind of memorization, old memorization, but you just look at look for the first aid for this. Okay. And then T cell development is also, for some reason, T cell development, it looks like the T cell development, the teacher doesn't like emphasize the T cell development, but I think this is also kind of important too. So what you need to know is the negative selection, then positive selection. So where the T cell made is always, all cells are made in bone marrow basically, right? So from bone marrow. And what T cell will be, after T cell is made, it has to go, all T cells go to the thymus. So this is a school. Okay. So yeah. Alex, yeah, really good job. Okay. So IL-1 is fever. Let me just bring that thing here. Here. Yeah. The one is hot fever, and then 2 causes a stimulus T cells. Okay, so I just send yeah, stimulus bone marrow alpha. Yeah, you can yes. So the problem was I told you about the proliferate, right? So stimulate T cell proliferate. The IL-3 IL-3 stimulates bone marrow. Okay, yeah, right. Okay, so memorize this thing. Okay, so this is a good, very good, the useful, the the useful the mnemonic. Okay, so now what happens is there are when the T cell is going, so T cell immature T cell like a baby T cell, right? Baby T cell, precursor T cell was the generated from the bone marrow, and then it goes to the school thymus, and then goes to positive selection and then negative selection. There's like a names, precursor T cells, naive T cells, naive cell post selection, that kind of things. You can just name it, but you can just memorize the names at the end. But basically you know this, try to understand the two mechanisms. One is positive selection, and then one is negative selection. Positive selection means that basically positive selection means that something you should do. Okay, so positive selection should do, and then negative selection should not do, not do. So what I meant is this. So T cell supposed to bind something you need to bind. For instance, suppose you need to bind, you need to so T cells job, all immune cells job is to kill the pathogen, right? Not kill yourself, right? So that's what, so kill should do means that the kill pathogen. So in order to kill pathogen, you need to bind something, right? Bind to the pathogen, right? The way you bind to the pathogen is through the MHC1 and MHC2. MHC2, MHC1, MHC1, and then the MHC2. Okay, so CD8 cell bind to the MHC1, CD4 bind to MHC2. So this is something. So the basically body will provide a kind of fake the MHC1 and MHC2. Okay, so hold it. I mean the tight, bind it, and then if the T cell bind it tightly, oh, you are good. You are selected. Okay, then you can go to the next level. You can promote to the next level. Okay, then now you do the negative selection. Now you give the kind of the self myself tissue. Okay, so myself tissue. Okay, so this is myself tissue. You should not bind this. You should not bind this too tightly. So then it will cause the autoimmune disease, right? So you don't attack me. You just attack pathogen, not me. Okay, so give them some kind of the. So this is kind of bait. Okay, bait. So you give them, okay, so give them myself, so piece of myself. Then, okay, do you wanna buy? And then if we bind too tightly, that's not good, right? So if we bind too tightly, then that's the negative selection. If you should not bind me like weakly, okay? If you bind too tightly, you're out. If you bind weakly, that's fine. Okay? So then you are now good to go. Now you are ready to go. You graduate from school, and then the becomes a mature T cell. Okay? That's what the process is. Okay? The corresponding names, once we pass this, what names, once we pass this, what names, yeah, you can just memorize at the end. Okay? So that's what the T cell, the maturation, okay? And then B cells, all you need to know is the B cells will have they have a precursor B cell, naive cells. Okay, those are kind of names that you need to know. But B cell, this important thing is that this the antibodies, antibody structure. Normally, I don't really try to focus on the anatomies, but this anatomy is very, very important. Okay, this anatomy is very important. So you need to know this all kind of the structures, names of these structures, and all these things. Okay, so basically you have this the constant region and then variable region. So variable region is only this green part. Okay, so remember that. Okay, and then constant region, and then also the constant region is like all other parts, right? And then important thing is that heavy chain and then the light chain. Light chain is this thing, So constant region has heavy chain and light chain. Okay. Then the what's important in the light chain is that when heavy chain and light chain supposed to be balanced, supposed to have about same the numbers, right? When the light chain is too much, what's going to happen? Not good, right? It's not good. Do you know any disease that has too many light chains as compared to heavy chains? Have you heard of the disease called multiple myeloma? OMA, right? The tumor disease, kind of cancer thing, right? Not really a cancer, but OMA, multiple myeloma is too much light chain. Okay, too much of this thing. Monoclonal, monoclonal means monoclonal means it has to have a like a multi-clonal. In other words, you have to have a many different versions of this thing, but only same thing will happen. Some same thing is many, not good. Okay, monoclonal. So monoclonal light chain overproduction is called multiple myeloma. Okay, so that's something you need to know. And then oh sorry, this one the B cell activation is it's actually important because the T cell is the T cell will present to the big cell, and then B cell will be activated, and then B cell will be the making the converted to the plasma cell, so it makes the like the antibodies. Also, at the same time, B cell will become memory B cell, so it will react more quickly when the next time the pathogen same pathogen comes in. Okay. And then, yeah, this is very important. Where does the pathogen bind to is also important. Okay, normally this is normal situation. Okay, normally pathogen is here, so like bacteria, and then this bind to the this this will bind to the bacteria. Okay, then FC terminal is bind to the kind of our immune cell. Okay, so let me just, yeah, immune cells. Immune cells means like neutrophils or that kind of stuff. Okay, B cells, T cells, that kind of stuff. Bind to here, and then here is bacteria. Okay, that's the normal situation. Some bacteria are super smart. So some bacteria bind to the FC portion instead of just there's a kind of some kind of receptor that when the antibody comes in, antibody is supposed to bind here, but then this bacteria has a very unique kind of the receptor that doesn't fit in here. It fits in here. Say I mean in this shape, it's like this shape, right? And whereas this is the kind of this kind of shape, right? So makes the and here's the receptor is like this shape. So it binds to the here this side. So then the when the neutrophil comes in, neutrophil cannot bind to this antibody, so cannot do the job. So this is very smart, right? So all kind of this the bacteria's smartness to avoid the our immune cells killing them is called virulent factors. Virulent factors. Virulent factors are very important when you study the microbiology. This is one of the big topic you need to know. Whenever they have a virulent vector, you need to know that. Okay, there are many virulent vectors, so you need to know this kind of things, virulent vectors. Okay, there are like the I mean the immunoglobulin types as I explained, IgM, G, A, G, D. Is there are 5 types. You need to know this to always IgM first. IgM is the first one to the generate, and then after that it will become like it will the plasma cell will the generate more. You need more the antibodies, then it will become IgM, then IgG, IgA, IgED. Right? Okay. Why the IgM is always the first one to be like produced? Look at this. It's got many binding sites. So it can bind bacteria here, here, many up to 10 bacteria. When you have a like new pathogen comes in, do you want to have like something detected bacteria in many different ways? That's exactly the same situation. So you don't want that, only few the policemen. If there's like okay, there's a shooting going on in there's a I say there's a big fight going on the street. You don't want to send the only one the policeman, right? You want to send a bunch of police squad, right? So that's what this is exactly. This is like a police squad. So group of many police will come in at the same time. Okay, so it can bind up to 10 bacteria. Okay, so that's why IgM is the first one to be produced. Okay, then IgG, and then all other things. The important thing is IgG crosses placenta. Okay, so when you deliver the baby, baby actually originally don't have any disease, right? So baby has like immune to everything, right? So no disease. So no disease means that he hasn't seen any the bacteria yet when he born. So uh so he cannot, because he didn't see any pathogens, he cannot make any the antibodies. He also, not only that, his immune system is not really well developed yet. But so the IgG, whereas it's kind of long-term, the long-term like memory. It has a memory, and then it lasts long. So the IgG God made IgG smallest as possible. So this smallest the antibodies can cross the placenta. So blood, baby will have the baby blood will contain a lot of IgGs. Okay, when he is born. Especially this one is very important when you even if you are not breastfeeding, the doctors recommend you to breastfeed at least 1 week after the birth. So colostrum, I think. Colostrum is the milk in first week, 1 week, less than 1 week. So as soon as you birth, the give the milk to the baby, because of that particular milk, very thick milk, is the kind of is kind of thicker than the regular milk. Actually, the breast milk is not very thick, but this one is super thick, very thick. Okay, for 1 week. Okay, so this colostrum contains a lot of IgG. Okay, so that's why the doctors recommend, even if you are not breastfeeding, feed the baby with breast milk. Give the breast milk to the baby for 1 week at least. Okay, so that's what that is. Complement pathway actually is an important thing, but uh, there's a classical pathway, IgM, IgG, lectin pathway, mannose, alternate pathway, many things, but all comes down to only one thing. That at the end, the C5 through C9 forms the MAC. Okay, MAC is basically death chamber. Okay, so that's what you need to know. And then there's a classical pathway is start from this one. This one start from C1 complex. Lectin start from C1 like C1 like complex. I have no idea what that is. Alternative the C3. You need to you just memorize at the end, but basically big thing is that it comes down to MAC. All pathways lead to MAC. Okay, so that's what you need to know. And then this is not really the unimportant topic. It is an important topic, but the what you need to really important thing is that you need to know this thing MAC, and then also what uh, complications is you have. This is an important thing. Okay. So paroxysmal nocturnal hemoglobinemia is a very important. It's a deficiency of CD55 and then the CD59. Okay, this thing forms the there's a red blood cell. It's a membrane, right? So this membrane is like some kind of this is membrane forming the the membrane forms, and then the complement will supposed to if there's something wrong, then complement will activate it, and then destroy the membrane. Now this thing is the membrane decay acceleration factor. DAF means decay accelerating factor. So it will, if there's a too much the complement, it will DAF will activate, so inhibit this thing, DAF. Okay, but if DAF is not is deficient, what's going to happen? This will be activated, and then this will destroy the membrane. Not good, right? So this is what happens in the paroxysmal nocturnal hemoglobinemia. The reason this is a very important is this is very popular the symptoms, very popular symptoms, and then the many people have it. And one of the good things you need to know is that you need to know this thing, and then why it occurs in the during the nocturnal hemoglobinemia, because only it happens in night. Why? Because in during the nighttime you have a high level of CO2 because you are not breathing very the like rapidly during the nighttime while you are sleeping. So, okay, so that's why it happens. Usually happens in the when the CO2 level is high, so you have more CO2 in the body, then it will kind of oh, there's something wrong in the body, so complement has to be activated, right? So complement activated, and normally this will be inhibited by DAF, but if DAF is not there, more activation here during the nighttime. So that's why this happens during the nighttime. Okay, so the symptoms is when you the morning in the when you go out and pee after wake up in the morning, and then pee becomes a sometimes like a red, but red is very rare. Usually thick yellow. Okay, thick yellow because the red blood cells died. Okay, so this is a kind of the how you diagnose this, and but really you're not diagnosing the because it's something to do with the antibodies. You diagnose the direct Coombs test. Okay, so that's something you need to do, right? And then active and passive immunity. Passive immunity means that, for instance, if I have a the like let me explain active first. Active means that your body makes the antibodies, and then the it will protect it will fight against the bacteria. Okay, but passive is that you bypass this, and then you just give the IgG directly. Okay? So it will be very fast, right? But it's like the it's like the when you are hungry, you give the food. Okay, so this is basically food, and this is like give you how to make food. This is better, of course. The active immunity is much better, of course. But in some cases, you have to this you have to be the passive immunity, because it's very rapid, because you don't have to time, you don't have time to cook. You just give the food directly. So you will be your hunger will be just resolved immediately. But you don't know how to make cook. You don't how to make food, right? So this is like very temporal, but sometimes is required, because if you need like immediate the treatment, then this is works very fast. Okay, so this is speed is the key. Okay, that's one thing. And where is it? Okay, and then there's a yeah, and then there's oh, what happened here? Why is it? Okay, and then there's two types of. I mean, there are many different types of vaccines. I think so. The what you need to know here is this. I don't think the you need to like they differentiate in many different ways. Okay, so all you need to know is live vaccine. When do you think the live vaccine will be contraindicated? When your immune system is, because of live is live. Live means that even it's alive. So that you give the like a small samples of the live bacteria. So if your immune system is very weak, you do not give this to the live bacteria. So immunocompromised people. Another the population that is contraindicated for live vaccine is the pregnant. Why is pregnant the contraindicated for live vaccine? Because pregnancy. What does pregnancy mean? Baby inside, right? What does baby inside mean? Baby is basically pathogen. Okay, let me write it down here. Baby is a pathogen. Okay, know this. You have to know this. Very big, important concept. Because the baby is a big pathogen in your body. So all of a sudden, your big pathogen, big bacteria coming into your body. So your the immune system, but the way it works is that your body try to not to kill baby, right? You don't want to kill baby, right? So what your body will do is, instead of killing the baby, because it's a pathogen, the normally immune system will come in and then kill the baby, right? Normally, right? So you don't want this happen, right? So what happens is the placenta and then baby and then the amniotic the fluid and that kind of things will secrete some kind of cytokines, so that inhibit the immune systems. Okay, so during the pregnancy, your immune level will be very low to protect the baby, to not kill the baby, okay? Not to kill the baby. Okay, so that's the important concept. Okay, that's why you don't give the live vaccines during the pregnancy. Okay, all right. And then another important concept is that conjugation. What does conjugation mean? Conjugation simply means adding something. Add something. Okay, so what's something? That something is a peptide, because T cells recognize only peptide. If there's like a carbohydrate or fat, they don't recognize this thing. Only peptide, because of bacteria, and then only all kind of organism will be composed of the protein, right? Peptide, right? So that T cell will be designed to recognize only the peptide. If there's a carbo, don't care. If there's a fat, they don't care. They cannot see it. Only peptide. So you make some other the vaccine, and then if it's not to like work better, this will be this vaccine is conjugated with peptide. Vaccine is conjugated with the peptide. So this is called conjugated vaccine. Okay. So that's something, that concept is something you need to know, right? And then there's also the new kind of new vaccine in the block, new case in the world, mRNA, Moderna, right? So this thing is a kind of new thing, okay? So that's something you need to know. That's for the COVID vaccine, okay? And then this thing, the immunodeficiency conditions, very, very, very important. But the this one is like maybe one or two classes. So I want you to be like focused on these classes. Very important concept, but it's too long to explain everything in the 2 hour session here. Okay, I'm already out of time, so I will just jump into the like hypersensitivity. Hypersensitivity is also, all you need to know is like the type 1. First exposure, it will let you sensitization. Sensitization means that the body will make the antibodies against this the exposure thing. Okay, so antibody means IgG or IgE. In this case, particular case, IgE. Okay, so that's what it is made. So in the first exposure, antibody is made, and then second exposure, it's ready to go, ready to attack this pathogen. The problem is ready to attack pathogen, but that will hypersensitivity, right? Hyper, not just type 1 sensitivity, hypersensitivity. So it will attack. It's supposed to attack the pathogen only, but in this time it will attack too much. So this the mast cell will be degranulated. So like granules will be come out. That will cause a lot of problems. This contains this the mast cell, the granules contains histamine, tryptase, and that kind of things. So all kind of inflammatory process will be happening. So this is the called type 1 hypersensitivity. Okay, so remember, first exposure nothing in terms of symptoms, because it will be sensitized, and then second exposure, the problem happens. Okay, anaphylaxis is one of the serious condition. Okay, type 2 is the type 2 is basically the something antibody. When the type 2 differentiating type 2 and type 3 is a blood, something to do with blood. So antibody and then antigen comes in, antibody and antigen combines and then circulating in the body, blood vessel, that's type 3. Antibody is not there. I mean, the antibody is not there. Antibody is just in the tissue, and then the antigen comes in. It will it will make the hypersensitivity condition. That's a type 2. Good example for the antibody attacking something is something is going to be our tissue. Okay, so good pasture is a one good example for the type 2, and then type 3 example is a SLE or the serum sickness. Okay, those are the things that you need to know. Right. Type 4 is delayed the hypersensitivity because it's a T cell mediated. So one of the good example is a poison ivy. I think this is a poison ivy, and then the poison ivy, and then Dress syndrome, the Stevens syndrome. That's something that's the examples are you need to know. They usually goes the examples. Okay, so acute hemolytic reaction. Okay, I'll just skip this one. And then because I wanna go for the micro. Oh, this transplantation is very, very important. So you need to know this thing very, very well. And the teacher will be also emphasize on this. But one thing I wanna mention, Charlie. Charlie is not very, for some reason, is not very heavily tested on step 1. but very, very important in step 2. I don't know why, but step 2 focused on literally very strongly. So although in the teacher doesn't really spend much time in trolley, but you need to know the trolley very well, okay? For step 2, not step 1, okay? All right. And then also hyperacute, acute, and those things. What's the important thing? Is a timeline. Timeline is very, very important. Hyperacute, I think it's less than 30 minutes or something like that. There's a chart in here. There's a chart, yeah. It's a very, very minutes. In minutes. So this, among these types, most important thing is that, this crash, the onset. Onset is very important, okay? Onset is important, and then of course, the mechanism is important. And so those two things are very important, okay? And immunosuppressants, this is like a really, really important thing. But I think that Sonny will be covering some of the pharmacological things. Okay, so um, so I told you about this, like natalizumab and those things, right? TNF alpha, adalimumab, that kind of things are called biologic immunotherapy. Okay, so this is very important thing, like this thing, adalimumab, and those things, TNF alpha inhibitors, right? So these things will cause TB. Okay, so any TNF alpha inhibitor, TNF inhibitors, is actually TNF alpha, same thing. Right? So TN will cause TB. The latent TB reactivation, because the caseating granuloma, which contains the live TB bacteria, okay? For the microbiology, I will just go over very, very quickly, because because the region 1, I don't have much time left. And then region 2, microbiology is basically, you cannot understand anything about the microbiology. Microbiology is basically all memorization. Okay, so purely memorizations. But I ask you to, I highly recommend to you do the watch the sketch. Okay, sketch micro is the way to go, because what happens is a sketch. Many people say that even in the teachers, they sketch micro is kind of too brief. So that's true, but the too brief, and then you need to know more than that. But my question is this: if you like sketch micro is considered as one of the standardized the materials resources. Okay, so let me put it this way: if it's not in the standardized materials, standardized resources, it will not be under step 1. Okay, so knowing sketchy, if something, some other information that which is not not in the sketchy, is it important? I don't think so. Okay, can it come out in the exam? Yes, it could be, but in terms of high yield, low yield, that will be low yield. Okay, so something you need to know, right? So so this is like a gram. That's basically gram positive and gram negative. So difference is that gram negative, gram positive has this like big cell walls. Okay, cell wall here big, but then gram negative has only small cell wall. But they have like a different layer here. Layers are here, and then yes. So this cytoplasmic membrane is the same, but this top layer, which is like outer, they have a gram negative has outer membrane, which has Taurine, I mean, that's not very important. LPS, endotoxin, they are basically the same thing. LPS, which can kill people. So whenever you see in some in the medical terminologies, which can kill people, you need to pay attention. Okay, this can kill people. Okay, so this is very, very important. LPS. So whenever there's something on the gram-negative bacteria attacking you, and you died, why? Because of this the endotoxin or LPS. They same thing. Sometimes some bacteria like a the Neisseria, the gonorrhea, Neisseria species, they have OPS, oligopolysaccharides. Okay, so that's the kind of variation of the LPS. Okay, so LPS will kill you. Okay, and then whereas gram positive, the lipoteichoic acid, and then which doesn't not have the this is unique to gram positive lipoteichoic acid. So if there's some after the infection by the gram positive, and then you have some kind of complication, that will be due to the the lipoteichoic acid. Okay, so there are all this is basically all these things in here are the weapons that the bacteria have. Among those, so like capsules is important. Capsules, if there's some bacteria that doesn't have capsules, many times uh, capsules are here, but some bacteria don't have capsules. That's important. So what bacteria is a shine my ski, I think. My ski. I mean, prism shine my ski. Sorry, prism shine my ski. So what I mean is the Pseudomonas and then Staphylococcus and then Haemophilus, the influenza and that kind of thing. So you need to know these things. Then Klebsiella. Those things in the first aid. First aid is the best for pneumonics. There's a Shinsky encapsulated bacteria. So it's called encapsulated bacteria. Okay. So that's something you need to know. Okay. So that's because when there's a capsule here, then what happens is the every blood has to go through the spleen, right? Yes. Who is this? Okay. So the I think it's in the chat. I think it's Alex, right? Yeah, Alex. Beautiful.
I'm
Yeah.
just
So this one.
summarizing, Kevin, so you don't have to like maybe they can read it later. Just whatever you
Yeah,
say now.
yeah. This is really good. Okay, this is a
Thank you.
really good. Yes, recommendations. Okay, so so every blood has to go through the spleen. Okay, spleen has a the spleen is kind of the water filter system. So all water coming to our house is filtered, right? So water filtration system is a spleen. So when the blood comes into the spleen, it will be filtered. In other words, it will be examined, then investigated, or if there's any the problems. Okay, the what they examine is the if there's any defect in the red blood cells, if there any the toxins, or if there's any bacteria, or if there is anything defect, then spleen macrophagy will just pick out and then kill them, eat up. They will eat up. Okay, so that's the spleen macrophagy job. When the spleen macrophagy will eat the bacteria, they will eat they will they cannot eat just open the mouth and then eat. They cannot do that. That's a human, but they don't do that. So what they do is they kind of everything in microbiology world is kind of it's kind of due through the like a receptor and the ion gate and that kind of channels and that kind of stuff. Okay, so that's like microbiology world, not like a human world. The eat up means that eating open the mouth and then they insert the food, right? So it doesn't go like this. So everything has to go through the receptor and the stuff, ligand receptor and that kind of stuff. But when there's a encapsulated, then this the ligand the gate channel is not going to work. Okay, so that's what the encapsulation means. So this bacteria, particularly this the Shinsky bacteria, please Shinsky bacteria, like Pseudomonas and those things, they cannot, if they comes in, spleen macrophages cannot eat them up, so they are free to go. So they have a so when this the when the those bacteria when it comes in, So they are the causing the problems. Okay, so that's the one thing. Very very popular topic and very popular the virulent factors. Okay, so you need to know that. All right, and then this is a good table for this gram negative and gram positive kind of the algorithms. So how to classify this thing. One thing I quickly summarize this thing is that there's a hemolysis. So hemolysis means that the breaking down the blood, right? So all this is basically rather than this is like clinically important. No, not really. Not, I mean, doesn't really matter. The what kind of the what kind of novobiocin, the boxytracin. It's not really clinically relevant, but very very importantly, they will be the very important tool to distinguishing what bacteria it is. Distinguishing because of that depending on the antibiotics or the say this this bacteria here is comes in, then you don't want to use the bacteria that is specific for this one, right? So you have to know what bacteria it comes in. So you can have the modify the bacteria. I mean modify the antibiotics to kill this bacteria, right? So these things are not very very important by itself to clinic in terms of clinically, but this is very very important distinguishing which bacteria it belong to, and then which antibiotic we will give to them to kill them. Okay. All right. So the alpha, beta, gamma hemolytic is one of the big thing that distinguishing this the streptococcus. Okay. Alpha hemolytic, so beta hemolytic is 100% hemolysis. Hemolysis. So when you petri dish, you give the blood here, and then beta hemolytic bacteria here. Then that will be all. This will be hemolyzed. Okay, whereas alpha is 50%. Yeah, alpha is 50%. So if you give the petri dish, it will be like this, hemolysis. Okay, the gamma is 0%. 0% hemolysis. So no, nothing will be the hemolysis. No, if you give the blood here, it will be just as is. No hemolysis occurs. Okay, so that's how you distinguish the alpha beta alpha beta gamma the distinction. Okay. So there are many there are other things like the bacitracin or the opticin and the kind of things, bile sensitive. Those are all kind of the you need to know like from the sketch micro. Okay, sketch micro. So I am big fan of the sketch micro. Right. Also the gram negative can be distinguished in different ways. All right. So TB mechanism, I explained this to you, and this will be on the exam for sure. If it's not on the exam, there's no way that this will not be on the exam. Okay, very 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 important. Okay, and this one, this the last pages, last few pages, last maybe 10 some pages is from the class note that PPT that the all depending on the like the specific the microorganisms. Okay, so you can just read through once. Not you don't have to memorize this thing. You just read through once and then watch the sketch. That will help you. Okay. All right, so time is up. So I'm gonna stop here, and then at 1 o'clock, the Alex and Sunny will be going over pathology and the pharmacology. All right. Okay. Any other questions? And I will see you. I think so. The next section, next day, I'm gonna do the micro. I mean the muscular skeletal and hemonco. Okay. All right. Thank you very much. Okay. Thank you. Bye bye. I'm gonna.
학생이 미국 사람이야?
I'm gonna stop here and then I'm gonna stop recording. Just a minute. Record, stop recording. And stop recording.